Okay. It's my pleasure today to introduce the speaker, Joanna Sulkowska, who is at the University of Warsaw. Joanna I have known for many years, and we've taken very complementary approaches to looking at knotting in proteins and other biological species. Joanna started her PhD looking at knotted proteins with Marek Chiplak in Warsaw, and then went to postdoc with Jose Anuchik, and has quite a few years now been running her own group at the University of Warsaw, and has done some wonderful computational work on knotted proteins and other species. And I will just hand straight over to Joanna.
Thank you for this wonderful introduction, and I should add that Sophie is somehow my mentor, that everything that I learned from the beginning of knotted protein, I learned by reading Sophie paper, and yes, without Sophie starting, I think I will not continue this. So thank you, Sophie. You are really my great mentor, and you introduced me to this field. So let me remind you that knots, we can say that knots surround us. We know that knots can be found in industry, and you can see this rope can be knotted, even if we can see this is the open chain. Knots can be found in, for example, in Africa. Here you can see two trees, they are linked. As well, knots are very common motif in art, and they are very beautiful. And as well, probably you know that there was a Nobel Prize in chemistry for designing so-called catenan. And here you can see two particles. They lead to discovery of rotaxane. And there was also the Nobel Prize in 2016 in physics that was awarded for theoretical discovery of topological phase transition and topological phase of matter. And the last one, you know probably very well from the previous talk, is the picture of knotted DNA. But what about the proteins? So here you can see a three-dimensional structure of protein. And first of all, I would like to ask you if you can see something special in the geometry of this protein. So usually proteins are characterized by primary, secondary, and tertiary structure, which describe the sequential structure, geometry, and in consequence determine the biological activity of the proteins. However, this protein has additional fascinating feature. It is entangled, and more precisely, we can say that can have a knot. To convince ourselves that this protein has a knot, we can, for example, pull it by termini and tighten. This is, as you see, this is very difficult to recognize the knot by just visual inspection, and this is one of the reasons I believe that knotted protein was discovered rather recently. However, today, due to many people, for example, Mansfield, Taylor, East, Birnau, Christoph Micheletti, and Sophie Jackson, we know that knotted protein exists, and we know as well that not only protein can make a knot, however, we can also find that protein can form lassos, and I will talk about the lasso later on. Here you can have a protein backbone that is closed by the cysteine bridge, and one of the tail is piercing this loop. And 2 years ago, we found out that protein also can make links, and the link is formed by the protein backbone, which is closed by two cysteine bridges. I hope you can see here there's one, and there's another one here. So this leads to many interesting questions, because on one hand you can say that 7% means a lot of structure, but on the other hand, this number is much lower than it is expected from the polymer physics. So why only 7% of protein deposited in the protein data bank are possess non-trivial topology? So how we can characterize such entanglement in proteins. Secondly, you can ask what is the biological role of entanglement. And the basic question, I think, that before we will understand the biological role of entanglement is we have to understand how knotted lasso and link protein can fold. So I think it's impossible to, this field is growing, so there is many people currently studying the knotted protein. So I only collect the information that I contributed to this field. And I can say there are some disadvantages and advantages for possessing the knot. So we can show that knotted protein are prone to aggregate. I believe they are slower folder. Their folding pathway is restricted. What is very important to notice is that knotted protein exists in the naturated state. So this is one of the reasons why we need such people like Sophie Jackson to be able to study this protein, because they are not easy to study experimentally as well theoretically, but theoretically, we can at least detect if protein chain is knotted, but from experimental point of view, it's very difficult to see if after the naturation, the protein is knotted or unknotted. And there is also some way that we can say that maybe it is difficult to degrade the knotted protein. And if you remember Sophie Jackson talk from the previous talk, she also mentioned that this is still somehow open question. Some knotted protein can be degraded, but not all. But there is also, I think, a little more advantages to possess a knot. So we know that knots, lasso, and links are very well conserved in protein, and I will come back to this topic later on. I believe that from the point of view, at least from the simulation, we can show that it's very difficult to degrade knotted protein, and they are more mechanically resistant than unknotted one. It is possible to correlate the lasso topology with some function, and I will talk about this a little later on. And I think at least in one case, we can show that knotted structure provide ideal environment to catalyze chemical reaction. So then you can ask why evolution conserve entangled structure. Or you can, so this is one of fundamental questions that we would like to know that will explain us why only 7% of protein are entangled, or this is a lot, or this is very small number. So how we can study the role of entanglement or the function of knotted protein? So this is not easy, but there is one protein that allow us to deduct something. So this is a methylotransferase. This methylotransferase possesses a deep knot, and it was explained later before by Sophie. Deep knot is a protein which cannot untied by the thermal fluctuation. So there is at least, let's say, 50 amino acids from N and C terminal before we will find the knot along the protein sequence. So this protein is a dimer. It's composed of 2 chains and is responsible for the modification of tRNA. This modification assures correct codon-anticodon pairing, and thus is essential for fidelity of protein synthesis and therefore for surviving the organism. So this protein is quite crucial for a cell to live. However, the same function is performed as well by unknotted protein that can be found in eukaryota and archaea. So we have two proteins that perform exactly the same biological function, but one is knotted and one is unknotted. They both bind exactly the same ligand in the active site. So I should mention, and it's very important, that the knotted protein bind the ligand in the active site, which is knotted, and this one bind the same ligand in active site. This is unknotted. So maybe if we will try to understand why evolution designed these two proteins, one knotted and another one unknotted, we can understand what is the possible role of entanglement. So I don't want to go to many details, but we can perform many point mutation, or it was done by our collaborator. And then we can find out that some mutation in the active sites give us surprising results. So, for example, this mutation in the position of 115, which is not directly responsible for binding the ligand or binding the tRNA, can lead to many modification. So why? So then we can, just to make the story shorter, we can determine the energy of binding as well from the simulation. And then we can try to study why this mutation is crucial. And when we analyze the simulation, then we can find out that mutation inside the knot indirectly leads to destruction of interaction of other amino acids inside the structure. So this can suggest there is some internal network of interaction that is network of interaction is controlling by something. And we believe that this controlling is the knot is responsible for controlling this interaction. So then you can ask why is a band conformation important for the function? I told you before that knotted protein bind the ligand in band conformation, while the unknotted protein bind the ligand in the open conformation. And we just to make this story shorter, that we can find that this band shape is the conformation necessary to conduct the metal transfer, where the open shape makes the metal transfer strictly impossible. So then you can ask the question if this band conformation is correlated to the presence of knot. And we were lucky to find that there was another protein that is, we believe that is supposed to be a knotted protein, but it was crystallized without the knot. So we can try to find is this band conformation can be bound to knotted protein that is by accident unknotted. And then we can find that this band conformation must be achieved in the knotted protein, but is not, but is energetically unstable in a unknotted protein. So we can conclude that only the knotted shape allow this protein to bind the ligand in the band conformation. So if this is true, then we can say that mutation inside the knotted core can lead to malfunction of this protein. And in also something what I did not mention before, we know that there is other protein that have similar function. They have low sequence similarity, but they can also be very well aligned, where you can see the knot here. So then we can conclude that in the presence of in proteins built like this protein, the methylation would not be possible without the presence of the knot. And why this is important? This is important because then we can think about designing some selective inhibitor that will only block knotted protein, so from the bacteria, and will not block protein that are unknotted one found in the human body. So then I would like to conclude that study the topology in protein is not only important from, let's say, mathematical point of view, but can be also very useful for our daily life if we are able to recognize the protein which are knotted and only block this one that are not really good for us, so the bacterial one, and not the human one. So let me, maybe I can ask now if somebody has a question, and if not, then I can talk more about knotted protein. There's any questions?
Can you hear me?
Yes.
Hi, so it's Sophie. I just have a quick question. So I'm really curious about this crystal structure of this TrmD, where it's in the crystal structure it's unknotted, even though it's supposed to be knotted. So do you think that the unknotting, that it never got to its final knotted state, or do you think it's somehow unfolded and unknotted during the crystallization process? Why
No,
do you think it's unknotted?
I think this is just the, if I can say, improper assigning the backbone. So we also analyze the density map of this protein very precisely. And there is a way that you can see that there is possibility that if you will connect this missing loop based on other knotted protein, then you can see it will be knotted one.
So is it just that the electron density is not very well defined in the region of the knot in that particular case?
Yes, because the loop is very flexible, and this was probably the reason why it was not really properly, the backbone was not properly assigned.
Okay, thank you.
So then maybe I can go with the update about the knotted protein. Okay, so today we know that majority of the knotted protein possess a trefoil knot. We have two different types of the trefoil knot, the left-handed and right-handed. We know that there is a protein with 4-1 knot, 5-2 knot, and 6-1. And what I can say more, let me see. Yes, so I can say that at least more around 1,000 protein deposits in the PDB are knotted. And we also can find that there is a protein with the slipknot that I will talk later on. And recently we make some new scan of protein deposit in the PDB with Peter Wiener, and we found that there is something very surprising. We found there is a protein that is composed of 3 domains, and only only the middle one is knotted. So you can see this blue color represents one domain. There is another domain that is a knotted one with a different color, and there's another domain that is with the pink one. So this makes the deepest ever knotted protein that we will expect. Secondly, we found there is also proteins in membrane. So up to now, we only know that knotted protein have rather globular shape. However, recently we also know that a protein can be knotted in a membrane. And surprisingly, we also find that the knotted protein can be found in mitochondria. So then you can ask the question how this knotted protein will fold, or it will fold before in the cytosol and will be transported inside the mitochondria through the narrow channel, or it will knot inside the mitochondria. And usually when we talk about the knotted protein, we connect the termini in infinity and find what is the probability that the protein will be knotted, and we call such knot a probabilistic knot. And we just focus on the main protein backbone. However, as you can see here, the proteins are much more complex. They can have a size group. And this size group, let's represent just by some different balls. And we reduce them to the protein backbone. So like what you can see here, and connect the termini. However, if we take into account also the size groups and different ions that can make the connection, then we can recognize that proteins can also make the probabilistic knots, including, for example, when we have the connection by the cysteine bridge, as you can see here. Could you see this? I think my mouse, oh yes, it's here. So this is the probabilistic knot formed not only by the protein backbone, but as well by the cysteine bridges. But surprisingly, if you will look a little closer, you can find that we can find the deterministic knot when we take into account the sulfide bridge. So I call this deterministic because the knot is exactly determined by the protein backbone and other covalent bonds that exist in protein. And this should be contradicted to the cysteine knots that are well known. However, I would like to remind you that this is not really a knot. You can simplify the structure and it will be a trivial conformation. But the true deterministic knot as well exists in the protein. So all this new structure are currently deposited in the new version of the knot prot that we continue to develop with my student Paweł Dąbrowski, Tumański, and Andrzej Stasiak, Eric Radman, and Ken Millet. However, I would like to also remind you that if we look a little closer on the protein chain, we can as well determine something what we call the slipknot. So sometimes ago we were able to design something what we call the matrix. This matrix allows us better understand the relation between function and protein complexity. So to create we represent the protein as a matrix, as you can see here, where one axis represents the distance from N to C and from N to C. So for example, from 1 to 156 and 100 to 156. Thus, each element in the matrix corresponds to different continuous subchain of the protein. So, for example, left corner corresponds to the full chain from 1 to 156, and the cell near diagonal corresponds to very short subchain of the protein. So everything what is here is just the protein backbone. So this allows us to determine minimal segment of amino acids which possess a knot, and cutting a few more amino acids from one of the termini when I tie a knot. So you can see here the green color indicate all amino acids which form a knot. So I hope you can see here. So the last amino acids, let's say that you can count before destroying a knot, it's 76 or 119. So this will be this point and this point. And if you cut a little more, then you will destroy the knot. But this also tells you how to find where the knot is located. So we can find this knotted core here, and then we can know if the knot is deep or shallow. And shallow knot will possess only a few amino acids on the N terminal or just a few amino acids on C terminal, and then the thermal fluctuation will be enough to untie the knot. Such representation of the protein as a matrix allows us to find that some proteins can have a slip knot. So the full chain is unknotted. However, if you will cut a few amino acids, let's say in this case from the C terminal up to here, then you can see that the internal chain from here up to here is knotted. So this is the way how we define the slip knot, and we follow the idea that was introduced before by Todd East. And if we represent the protein by the such matrix, what we call the fingerprint of the protein, then we can recognize that, for example, this protein possesses internal knots. We can see there's a triple knot here. If we cut a little more from the C-terminal, then you can see there's a 4-1 knot. And then we can see there's another 3-1 knot here. So now I would like to tell you something about the folding knotted protein. But before I will talk about this, then I would like to remind you what we know about folding protein in usual. So we usually say that protein has a rather smooth landscape that was introduced by José Onucik and Peter Wally-Ness, and we can say that protein that cooperativity and increasing degree of nativeness are required for rapid and efficient protein folding. So we say that the landscape possesses some narrow or small traps on the way that are not enough deep to trap the protein, and the protein is able to fold to the native state. However, in the case of the knotted protein, this is not so easy because not all possible conformation are allowed. And there's a many other interesting questions. You can ask how biology, how to tie a knot, and that this protein will be knotted. So some times ago, I followed a nice paper of Sophie Jackson that she proved that it is possible to fold a knotted protein, but it's somehow not easy. And we construct the structure-based model with José Onucik, and we find out that indeed it is possible to fold the protein. However, we do not observe the random knotting, but instead we observe very precise pathway, that the protein will make a native twisted loop, what you can see here. So this is almost the native conformation of the protein. And then the one of the tail has to be dragged across this native loop, but it's dragging not just by pushing the terminal inside the loop, but instead we are doing something what we call the slipknot conformation, the same one that I explained you before. And then we can see that it is possible to tie a protein. However, we are not very successful. We only observe this successful folding into % cases, and all other unsuccessful trajectory lead to some topological traps. However, I should mention that it's also possible to fold this protein much more efficiently if we add no native interaction. So from this we conclude that it's possible to fold the protein. We do not observe random folding, but very precise pathway. But still there is some hidden mechanism that we need to find out how the knotted protein can fold. However, surprisingly, with Peter Villeneuve, we found that we can use exactly the same pathway to fold the biggest, this time, knotted protein with the 6-1 knot, just by what it was suggested as well by Taylor, making a twist loop and then dragging one of the terminal in the slipknot conformation. So we can fold this protein. However, this is also a very small number. So then I would like to remind you about two very good papers from Sophie Jackson. She was able to show that it is possible to fold a knotted protein, the same one that I discussed before, and probably the chaperone will facilitate folding. And additionally, Sophie made another very nice experiment when she attached two bulky or two stable proteins to the both terminal of this protein, and we also can observe probably folding in this case. So then what is the most important, we can conclude that proteins can knot in the cell-free system. So because we can still not observe the knot from the experiment, what I understood, but we can see what is the final result, then we can use this protein with the fusion to study the deepest knotted protein. So the fundamental question is how this protein with very, very deep knot can fold. So there's at least two possibilities. We can have the random knotting mechanism that we will expect from the polymer physics, or we can have a native twisted loop mechanism that I was suggested before, that we have a twisted loop, and one of the terminus of the protein will be thread in the slipknot conformation across this loop. Or we can think about another possibility. We can think about the formation of knot exactly when we come from the ribosome. So let's test all of these three possible cases. So we can, for example, compare these results to the results that from Sophie Jackson and to this protein that was fused to this domain. And it's also maybe unfolding this protein will help us to understand some biological meaning of this protein that we still don't know how it will fold, and maybe this will have some medical application. So which of these mechanisms is possible, at least from the theoretical point of view? So we tested this random knotting mechanism, and we will expect that we have a sufficiently long chain. We should be able to randomly knot this protein. However, we find out that it's impossible. So we ran many simulations, and we did not observe that even 2% of cases we could fold this protein. So then we make another theoretical experiment. We make a shallow knot, what you can see here. So we can make this shallow knot by, for example, heating the protein in very high temperature and see how the knot will move and generate this conformation. Or we can have the protein with a deep knot and ask the question if it's possible to fold now this protein if we already have a knot. So we found out that if we have a shallow knot, this knot will almost always untie. Before the and it will, if it will untie, then the protein will collapse and it will not reach the native state. However, if we start with a deep knot, so we will have, as you can see here, we finish with the 60 amino acids from one of the terminus, and here we start from 70 amino acids already. If we start from 70 amino acids, we can have very high probability that this protein, the knot will stay, and the protein will beautifully fall to the native state. So this is telling you that if we have the shallow, if it will be possible to make at least shallow knot with the random mechanism, then we should be able to fold this protein. However, we are not able to observe this. But if we will have a deep knot, then we can fold this protein. So if you will denaturate the protein and the knot will be along the chain, then we can easily fold back this protein. So we will conclude that this random mechanism is probably not possible, at least from a theoretical point of view. So then let's try the native twist loop mechanism. Okay, I was too fast. Let me come back. Yes, so what about the native twist loop mechanism? So we find out that we cannot fold this protein in such case. A short way to say this. However, it is possible to cut the C terminal and as well to cut the N terminal. So have only the middle domain, and this domain is stable. And what we found that if we have only the middle domain, then we can easily fold this protein and make a knot. And surprisingly, we observe also the folding via the slipknot conformation. So if we will only study the knotted domain, yes, indeed, it is possible to fold this protein without any additional help from no-native interaction, for example. So we conclude that native twist loop mechanism is also not possible here. So let's test the last possible chance, the ribosome mechanism. So this is the picture of the ribosome. It's a little more complicated, but here you can see RNA that is making finally the nested polypeptide chain, which should knot finally to make a native knotted protein. So we can represent this ribosome in a little more simplified picture. What is important for us is this ribosome channel, what you can see here. I hope so. I do not see my mouse. Oh, so here you can see this ribosome channel. We know what is the diameter of this channel. And what we can do, we can try to simulate the folding of the protein directly on the ribosome. And we can just represent this complicated ribosome as a tube with the diameter that is exactly that it's known from the experiment, and we can try to see what will happen. So the best way maybe to show this is a movie. So we constructed such ribosome, and then we push this protein outside of the ribosome. And what you can see here is just the protein nested chain coming is unknotted, and green part corresponds to the part that it should be knotted. So what you can see here is just the green part will make the loop, and this loop we attach to the ribosome because we know there is some interaction. And then we observe what's happened later on. And then what you can see here is that the ribosome is pushing the C-terminal across this native twisted loop and make exactly a knot. So if we release this interaction, then we can see that we nicely form a protein with the deepest possible knot. So this is a way that I currently believe that protein can be knotted, maybe. So honestly, I should say that the mechanism that we suggest before for the other protein with not the deepest knot, unfortunately, will not work out here. This new mechanism allows to folding, let's say, any protein independently of where the knot is located, because it doesn't matter if it's shallow or deep. If we make this twisted loop, then the ribosome force pushing the chain can just push the C-terminal across the native loop. And in this case, we observe successfully at least 60% of cases the protein cannot. So maybe I can ask if there is some question to this part. Sophie?
Hi, Joanna, it's Sophie again. You knew I would ask lots of questions because yes, I'm really fascinated by it all. So the interactions between the nascent chain and the ribosome that you introduce, are those specific interactions? I mean, are there particular residues that you think are involved in those interactions?
Yes.
Are they kind of bulky hydrophobic residues?
So no, we scan all possible data that we find about the interaction between the proteins and the ribosome. And we also scan the sequence of this chain. There is some server that can suggest where there will be attractive interaction between the protein and the ribosome. So we use this another server to choose this amino acids that will have the strongest interaction with the ribosome, and we just use the attractive soft potential to attach this 4 amino acids with the highest probability that will interact with the ribosome.
And have you looked at the sequence of proteins which don't become knotted and shown that they don't have these interaction sites?
I think so we did not make the general or let's say the comprehensive analysis of all possible protein, but it is a very good idea to make a distribution of such interaction. But I think any protein could interact with the ribosome. It's a question if this interaction will help knotting or it will just help attaching protein to the ribosome before protein will diffuse from the ribosome.
Yeah, I was just wondering whether it meant that some nascent chains of unknotted proteins ended up knotted.
So I could think that this could be possible, but if it will be knotted, then probably this protein will not be correctly folded.
Yeah.
So it could go to the pathway of the degradation, I would say.
Yeah. Yeah, but that might happen.
This is something what we cannot really observe because such protein will be degraded before we can catch this.
Yeah, it would be hard. It would be hard. It wouldn't. I don't think it would be impossible, but you could keep the chains attached to the ribosome. But it would be a difficult experiment to try and capture an aberrantly knotted nascent chain. But anyway, I like the idea, and I should let you continue.
There is also what we are trying to do now is trying to test your protein, and let's see what we can observe from the protein that you studied before.
Great.
Okay, so should I continue or there's other questions?
No, not from me.
Okay, so let's continue. So now I would like to tell you something about the Lassos proteins, which we find just 3 years ago. So in this case, you can have a protein backbone that is connected by the cysteine bridge. And then you can ask the question, what is the probability that one of the tail will pierce this loop? So to scan the PDB and our protein data bank and see if such protein exists, we use the approach what is called the minimal surface, and then it was applied in mathematics a lot of time before. And so here you can see also application of the minimal surface to art. That's, I think, it's a very nice picture. So if we use this minimal surface area and the triangulation method and span this minimal surface on the protein backbone that is close by the cysteine bridge, then we can see if at least one of the tail pierce the surface spanned on the protein backbone. And because we also can just easily calculate the vector products, we know from which direction this tail will pierce the surface. So if you will apply this method to all proteins deposited in the PDB, then we found out that at least 18% of protein possess non-trivial topology, which we call the Lassos. So we can have the protein with one single piercing, and we call this single lasso. We can have also the protein where the tail is piercing the surface or the loop twice, and we call this double lasso. We can have the protein with 3 piercing. But what is the most surprising for me, we can also find the protein that is piercing the surface at least twice, but from the same direction. So you can see here that I am coming from below the surface, then I am coming back and again below the surface. So we call this supercalling because it reminds us the supercalling in DNA. And in this case, if you will pull the tail, you can just untight maybe the structure. But in the case of the supercalling, you can never untie the structure if you will not break the cysteine bridge. And because the protein can have two tails, two terminis, then you can see that there is also the surface can be pierced also two times, but by two terminis. So then, because protein can have the left and C-terminal, and we can also divide this lasso a little more precisely. So you can see that the surface can be pierced from below the surface, but it's the same thing can be pierced from the top. So we call this L1 plus if it's from below N-terminal, or by minus if it's above. But the same situation can happen in the case of the C-terminal. So if you will summarize all protein with the Lasso topology, then of course you will expect the most common one are with the simplest topology. There is almost no difference from the statistical point of view, between piercing the C-terminal or N-terminal. But there's surprisingly difference when we have much more proteins, when we have this negative piercing, not the positive one. And the most typical, because we know which amino acid is piercing the surface, then we can see what is the most typical distance from the cysteine bridge to the moment then the tail will pierce the surface. So this is around 10 amino acids. So then we can ask a question: if we observe all possible type of lassos in proteins, and here with the green color, with the blue color, you can see the lassos that we observe in PDB. So all possible combination with one piercing. There's also almost all possible combination with two piercing. So it can just the double piercing, or two tails can pierce. And we can have also supercalling, but only from C-terminal, but we do not observe structure when we have the supercalling with the piercing from N-terminal. However, we can have also tripping piercing that you can see here. And in this case, you can see that definitely not all the structure exists, but surprisingly, we do not observe lassos with 5 piercing, but we observe lasso with 6 piercing. So this can be with L6N. So this is N-terminal piercing this loop 6 times. This 6 piercing can be formed as well when we have one tail that is piercing 4 times and another is piercing 2 times. And surprisingly, we have also the structure when we have the piercing from both terminals with that is pierced the surface finally 7 times. So then you can ask the question if we will observe more complex structure, and what is the probability that the lasso will be formed in the protein. So something that should be studied, I think so. And I mentioned before that we can correlate as well the function with the type of the topology. So it's possible to make the correlation that the simplest type of the lasso is found in the antibacterial proteins and toxin. And I should mention that this lasso peptide, not lasso protein, was found before, and it was already proven that, as you can see here, that we have a lasso peptide that is close by the covalent bond, and one of the tail is piercing this chain and this loop. We have 2 bulky amino acids. Hope you can see this. Where is my mouse? Oh. So you have 1 bulky amino acid here and 1 bulky amino acid here, and these bulky amino acids perfectly block the C-terminal in this case before threading from the loop. So definitely this topology provides the stability. And it's worth to mention that this, for example, the most complex structure is as well found in cell adhesion. What is the function? I don't know, but there is there should be some function, I believe, because they are very well correlated. So let me just move to the last topic. I already mentioned that we found links in the protein, but I would like to tell you how complex links can be found in the protein. So this is, for example, the protein backbone or protein chain that is reduced just to the protein backbone. And you can see that there's a cysteine bridge. So here you can span this minimal surface. This is how we found the Lassos proteins. And then you can see that one of the tail is piercing the surface. However, you can ask the question, what happens if the tail will be a little longer and will make another closed loop that it will be closed, for example, by the cysteine bridge. So in this case, you can see that we have two loops that make a link. And this is the way how you can scan all existing protein in protein data bank to see if such topology exists in the protein. So you have a protein backbone that is closed by the cysteine bridge, make the first red loop, then you continue with the protein backbone, you make another cysteine bridge, which close the blue loop, and then you're going to the end of the C-terminus. So if you know where is the position of the cysteine bridge, and you know that you span the minimal surface, then you can see that one loop is piercing the another one. So if we use this information and scan all the PDB, then we found out that indeed protein can make links. They are rather simple, but we found two types of the Hopf link according to different chirality, so it's pierced in different way. And the most complex one is a Solomon link. So then you can ask if this link is, because in the case of the knotted protein, you can have many point mutation and it will not destroy the knotted structure. But in the case of Lassos and links, just one single mutation of this cysteine bridge can destroy this topology. So then you can ask if this cysteine is conserved along the sequence, and we find out that it's very well conserved. So almost we did not find any cases where this cysteine bridge will be not conserved. And additionally, you can cluster or find what is the similarity between different proteins. And if we cluster all protein with the links according to some sequence similarity, we can find that links, the Hopf link is represented by 14 different families or class, and the Solomon is only found in 2 families. So they are very well conserved. And additionally, what you can see that the L1 is the size of the loop. So the first loop is rather small, and also the second loop is rather small. Only in 2 cases we found that the loop is rather big, and in such case we will say that there is no probably correlation between this topology and function. However, in these cases we can expect, because the loop is so small and the distance between the loops is also very small, that maybe this topology is providing some function or advantage for the protein. And additionally, we know that cysteine bridge usually provide the stability. And here you can see that all this protein can sustain very high temperature. So then you can ask the question based on one of this protein that it was studied before, and it was shown that it's very difficult to fold this protein, and there is a lot of problem with the degradation. What is the function of this topology? So then we performed the theoretical denaturation of this protein, and we studied 5 possible cases when we do not have the any cysteine bridge. So we have the trivial topology, so no cysteine bridge, trivial topology. We can have just one loop and one cysteine bridge. We can have only one loop, but just the blue loop closed by the cysteine bridge. But what we can also construct, we can construct the protein with the two cysteine bridges, but still trivial topology. And finally, we can have the structure with non-trivial topology, but the same number of the cysteine bridge. And we can measure the energy that we need to unfold this protein. So let's say this is the energy that we need to overcome to unfold this protein when we have just the trivial structure without any cysteine bridge. Of course, if we add one cysteine bridge, then the energy to unfold the protein is a little higher. When we have the just red loop, the energy is also higher, but there is almost no difference if it will be the red and the blue loop. However, what happens if we will have trivial structure and two cysteine bridges? So you can see that, as you will expect, that the energy to unfold the protein is much higher. And so the final question is what's happened if we will have the same number of the cysteine bridge, but not trivial structure. And what you can see here, the energy is higher. So then you can see that the Hopf link structure has the energy barrier over 20% higher than the trivial topology. And I think this is showing that this pure topological effect may explain the biological advantage of links in protein. So just non-trivial topology provides the energy barrier to unfold this protein higher. So I should mention, and I would like to mention, that it was the simulation and the idea is coming from Paweł Dąbrowski, that we studied this 2 years ago. So just to conclude, I think this non-trivial topology provides the advantages for the protein structure. The protein is much more stable. However, it's also providing disadvantage because, as you can see here, from the topological point of view, there is only one pathway that leads this protein to correct folding. I hope you can see this. So this is this pathway on the here. It's coming from the slipknot conformation that can lead you to the correct folding. But there's many traps on the way when we make the incorrect loop, or the loop will be just too small to thread the terminus. So the evolution has to solve this problem. What is better, to have easy way to fold or to have more stable conformation? And then I would like to just finalize with saying asking, are there more complex links? So we can have the protein with two cysteine bridges, which make a link, but we can have also two proteins that can be linked. And you can also ask about the probabilistic links, as well in the case of knotted protein. So we can have two protein chain that can be linked, but we will call this the probabilistic link because we have to close the terminus. And then if you will have this probabilistic approach, then you can see there is many protein in protein data bank that with some probability will be linked. So with this 50% of the chance, then we have just the Hopf and Salomon link. However, if we lower the probability of closing the chain, then we can have much more complex structure. And then you can finally ask, are there more complex non-trivial topology? If you will take into account size group in the protein, then we recently find out that protein can also make non-trivial theta curves. And that is also, I think, very surprisingly, and it was not investigated before. So today I would like to say that we have at least 8% of protein in PDB. There are entangled. We have knots, links, lasso, and theta curve. The structure are very well conserved in proteins, and topological analysis is important to fully understand protein properties. And if you would like to find such structure, you can can go to the knotprot. We have already the knotprot tool, which collect all information about knots, but as well we included the knotoids that are developed by Andrzej Stasiak. You can also look for the lasso and links in the linkprot. And if you would like to construct your Lassos protein or link, we designed 2 plugins that you can easily download these plugins and also form Lasso. So this is, for example, the protein where you can span the minimal surface, then you can smooth the chain and you can see where the lasso is located. As well, you can introduce the cysteine bridge by yourself and see if such structure will make a lasso. And the plugin allows you to study the protein as well, just the polymer chain. And the same with the links. You can, in this case, you can also make the links between by the cysteine bridges or by the ions. And then you can span the minimal surface and see what type of the structure you can observe. And with this, I would like to thank you. And I would like to mention all people in my group because everybody is important, but I would like to give the credit to Paweł Dąbrowski and to Mański for design or finding the links and theta curves. And Maciej Piejko was involved in the ribosome study. And I would like to thank you, all my collaborators, and mention also the Elinor Hagland with point out the Lasso protein for me when I was still a postdoc in José Onucci group. Thank you.